Welcome Junior Takis, my name is Mrs. Ramlal. This video I have prepared for the grade 12s to help them with their final exam preparation. In this video I'll be looking at the Gauteng Prelim 2017, the paper 2. Before you watch the video, make sure you have the question paper and the memo at hand. Right. 1.2. Give the correct biological term for each of the following descriptions. Write only the term next to the question number in your answer booklet. Right. 121. A large opening at the base of the skull through which the spinal cord passes that we know foramen magnum. Look at how foramen magnum is spelled. 122. The type of inheritance where two different alleles of a gene are expressed in the phenotype. Two different alleles are expressed in a phenotype. We know it's codominance. 123. An explanation of evolution that describes the speed at which it takes place. So wherever you say the speed at which evolution takes place, that is punctuated equilibrium. 124. A genetic cross involving one characteristic, that is a monohybrid cross. 125. A testable statement that can be supported by scientific evidence, that is a hypothesis. 126. The natural shape of the DNA, a double helix, not a helix. And 127, the position of a gene on a chromosome, a locus. And 128, a sex-linked disorder that affects the photoreceptors in eyes, color blindness. Right, 1.3. Indicate whether each of the descriptions in column 1 applies to A only, B only, both A and B or none of the items in column 2. Write A only, B only, both A and B or none next to the question number. 131. The physical appearance of an organism due to its genetic composition. When they use the word physical appearance, that is definitely phenotype only, so your answer is B only. 132. A sex-linked disorder. From these two, which is a sex-linked disorder, it's only hemophilia. Down syndrome is not, so your answer is A only. Right, this is a phylogenetic tree. And there are a whole lot of questions based on the phylogenetic tree, but make sure you read the phylogenetic tree carefully. From here, there's 6 million years to present. 141. Name the most recent common ancestor of the Homo genus. So you look for the Homo, and here they are. Now we look for the most recent common ancestor. So we follow, and there we are here, Australopithecus africanus. When you write this down, the A must be a capital A, and the Africanus a small letter a, and you underline them separately. As you can see, it's in italics, so it's a genus and a species name. Right. 142. Give the period of existence for the species mentioned in 141. So they want how long did the Australopithecus africanus exist. So if you look here, we're going to look at this line here. And so if we take that line, that is about 2,8. The memo also accepted 3 as well. So you could have read it from here as well. Till here, which is about 1 million. It would have been, I know in that year we accepted a bit of variations, because now I'm working on the computer, you will work on the paper. So, and you always have million years ago. 143. State the number of genera represented in the diagram. Now, the number of genera is, you look at the first name. So here's Pan, 1, Homo, 1, Paranthropus, 3, Australopithecus, Ardipithecus. So 1, 2, 3, 4, 5. 5. There are 5 different genera. So with genera, you look at the first name, and that's how you know what a genera is. 144. Based on the diagram, give one facial feature that differentiates humans from Australopithecus africanus. So you have to look at the humans, and you have to look at the Australopithecus africanus and give one facial feature. And you have to look at the diagram. So you can see they have flat faces, while you could say Australopithecus africanus have sloping faces. Humans have no eyebrow ridges, or you could have said Africanus has eyebrow ridges. Humans or Homo sapiens have a developed chin, or Africanus no chin. Homo sapiens have, or humans have not prognathous, while Africanus is prognathous, and you could have spoken about the cranium size as well. So there you wanted one. So just mention any one. Right. 145. Identify any two species that used both tools and fire. Now, we're looking at tools and fire, so here is where we're looking at. So anyone in this range, so you could have said Homo erectus, you could have said Homo sapien, Homo neanderthalensis. So as you can see here. And then 146. Name when any one Australopithecus africanus fossil found in South Africa. That's just based on your knowledge. So it could have been Littlefoot, Mrs. Place, Tom Child. Again, they marked the first one only. Right, 147 is not based on the diagram, so it's easy for us to answer. 147 lists two types of evidence that can be used to support the Out of Africa hypothesis. Well, of course, we're going to use fossil evidence, genetic evidence, or mitochondrial DNA, or cultural evidence. Again, we mark the first two only. Right, 1.5. In fruit flies, the characteristic for body color is either gray or black. A second characteristic is normal wings or vestigial wings. Vestigial wings are crumpled and therefore prevents the flies from flying properly. 151. In a cross between a fly that is homozygous dominant for both traits and a fly that is homozygous recessive for both traits, all the offspring are gray with normal wings. Identify the dominant trait for body color. Now it says dominant trait for body color. Can you see all the offspring are gray? So what's the dominant trait? Gray. Then they ask you to identify the recessive trait for wing type. Of course, recessive trait for wing type is vestigial. Okay. Because here all of them are normal, so the recessive one is obviously vestigial. 152. This is a cross describing two characteristics. What type of cross does this represent? Of course, a dihybrid cross. Then 153. Use the letters big B for body color and big W for wings. A fly is heterozygous for both traits. Give the genotype of the fly. So it'll be big B, small b, different letters. Big W, small w. So that's how you write the genotype. What will the phenotype be? Big B is obviously for, let me just look at the key. So big B is a gray body and normal wings. Right. And then they ask what's the possible genotypes of the gametes that this fly produces. So this is how I work out my gametes. I use foil. So big B and big W. Outermost, big B. Sorry, let me just fix that up. It does not look very clear, so it's a small w. Then innermost, BW, and the last one, BW, as you can see there. Okay, so that's how I work out the genotypes of the gametes that this fly can produce. Right, 1.6. The table below shows DNA-based triplets that code for various monomers of proteins. 161. Where would the DNA template be found in the cell? In a nucleus. 162. Name the monomer X. Now, that's obviously, can you see there's names there? Phenyl, alanine, cysteine, serine, tyrosine. That is an amino acid. Name the bond that would form between the monomers. The bond between proteins is a peptide bond. Then they ask you, give the codon for tyrosine. So it'll be ATA is tyrosine. They ask for the codon, which is the mRNA. So it'll be UAU. Okay. Right. So that's how we work it out. Then they ask for the anticodon for cysteine. So ACA is the DNA. Then you change it to the mRNA, which is UGU. And then you change it again to the tRNA, which is ACA. So I know it's a long way, but at least I know my answer will be correct. It's a final exam. I don't take chances with marks. Right. 2.1. The graph below shows the variation of heights of a group of 18-year-old students. 2.1.1. Identify the type of graph represented. As you can see here, the bars are together and there's no spaces, so it's a histogram. 212. Explain why this type of graph mentioned, a histogram, is more suitable for plotting data on continuous variation because it shows a variation of a range of measurements that are linked or connected. Okay, what is the most common height? As you can see there, 1.6 meters. How many people in the group were shorter than 1.3 meters? So, shorter than, so how many is that? So it's here, it's about 4 and 1, so 4 plus 1 is 5. Right, 2.2. Insulin is a protein. The diagram below shows the process of how insulin is made. 221. Name the following molecule X. So here's molecule X. Can you see it's carried an amino acid, so it's transfer RNA. Right. Name the following process B. Process B is taking place by the ribosome, so it's translation. 222. Describe process A. Process A, of course, is transcription. So how do you describe it for 5 marks? The double strand of the DNA unwinds. When the hydrogen bonds between the two strands separate or unzip, one strand is used as a template to form the mRNA. This is called transcription. Using free RNA nucleotides from the nucleoplasm, the mRNA is complementary to the DNA. This you will get in the exam guidelines. Right, 223. A sample of DNA was analyzed and 28% of the nucleotides contain thymine. Calculate the percentage of nucleotides which contain cytosine and show all your working. Right, so there's 28% of thymine. Obviously, thymine and adenine are exact numbers, so 28 plus 28 is 56. Now, the remainder, 100 minus 56 is 44, and then you divide that between cysteine and guanine, which is obviously 22%. Nice question. It comes out a lot in the exams. Make sure you write this down and make a note of it. 224. Describe how a DNA mutation will affect the structure of the protein formed. Now, this is just a general answer. The sequence of nitrogenous bases on the DNA will change. This will cause a corresponding change on the mRNA of the codons. A different amino acid will be brought in the tRNA to match the changed codon. This will lead to a change in the sequence of the amino acids in the polypeptide chain, and a different protein will form. And then 225, tabulate two differences between DNA and RNA. Remember, tabulate means to draw a table. So with DNA, you'll say the sugar is deoxyribose, there's pairing of bases, has weak hydrogen bonds, it's a long chain, it's double-stranded, and has the nitrogen base thymine. RNA, sugar is ribose, no pairing of bases, no weak hydrogen bonds, short chain, single-strand molecule, and it has a nitrogenous base uracil. So you will have the same idea opposite each other. So they talk about sugar for the one, and then the other for ribose, you also have the sugar mentioned in the same line. Right, 2.3. The diagram below shows a cell during a phase of meiosis. 231. Give labels for A. A we know is a centriole. Give labels for B. That is a homologous chromosome or bivalent. 232. Identify the phase in the diagram. Can you see the chromosomes in the middle? And they're in pairs, so it's metaphase 1. Then they ask you 233, give a reason for your answer. Homologous chromosomes are present and the chromosomes are lined up at the equator. 234, explain why this is a diploid cell. Can you see? Because there are pairs of chromosomes, homologous pairs of chromosomes are present. 235, how many chromosomes will there be in each of the gametes produced by the cell? So we started off with 4 and at the end it will be 2. Right. And then 2.4. Explain the importance of genetic variation in the process of evolution. Genetic variation causes variation in the phenotype or characteristics of organisms. Some are better suited or some have favorable characteristics as reference to natural selection of more suitable organisms. The sexual selection of different phenotypes in different environments eventually leads to species changing or change in the gene pool or change in gene frequencies. Grade 12s, I would write this answer down and learn it. 2.5. The passage below refers to genetically modified foods. Genetically modified food on the menu. A research agency survey has found that South Africans are becoming more accepting of genetically modified food. The survey found that 64% of South Africans now believe that GM food will become more widely accepted and less risky in the next few years. An earlier survey revealed that people thought the risks outweighed the benefits. 251. Define the term genetically modified food. What is GM food? It's the manipulation of genetic material of an organism to get the desired characteristic, or it's a process where genes on the DNA are changed or manipulated to produce a new organism. 252. Give 2 steps that should be consistent in planning the survey. When you're planning the survey, select specific questions to address the question, arrange the time for the survey to take place, design a table to record results, select suitable areas for the survey, and identify a sample that is representative of the South African population to question. 253. State the following with regard to genetically modified food. They want 2 benefits. So benefits: production of medication or resources are cheaper. It can control pests with specific genes insert into a crop. You can use specific genes to increase crop yields or food security, and you can select genes to increase shelf life of plant products. Then they're asking for 1 disadvantage. It is expensive. The research money can be used for other needs. It's interfering with nature, affecting biodiversity. It's immoral. It's against God's will. There could be potential health hazard and unsure of the long-term effects. 3.1. The diagram below shows the inheritance of coat color in pigs through 3 generations. 311. What type of inheritance is shown in the diagram? So can you see here is red, white, and pink? So of course, I definitely know it's incomplete dominance. Explain your answer. Why? Because some pigs have pink coat color. That's a third or a new phenotype. And neither red or white is dominant in this diagram. 313. Use big R for the allele for red coat and big W for the allele for white coat color to give the possible genotypes of individual 1. Individual 1 is red, so he has to be big R, big R. Individual 2 is pink, so he must be big R, big W. Then 314. A pig farmer is interested in breeding pigs with red coats, so he has a red coat male pig that he wishes to use for breeding purposes. He crosses the pig with individual 3, which is a pink coat female. Use a genetic cross to show the probability of the piglets born with red coats. So, red coats. So there's your key, red coat male. So you have P1 phenotype, red coat male, pink female. Then big R, big R, big R, big W. There's your gametes, you've written it. And what's your offspring? Big R, big R, big R, big W, big R, big R, big R, big W. So it's a 50-50% chance. And that's what they want to know. It was a 50% chance of them having red coat. Make sure you have parents and offspring, P1 and F1, meiosis and fertilization. And can you see here? This is the compulsory mark, meaning you had to answer this question here. Okay. Right. Brassica plants have hairs on their leaves to reduce transpiration. The number of hairs on the leaves vary from plant to plant. An investigation was conducted to determine whether artificial selection could increase the number of plants with more hairs on the leaves. The students carried out the following procedure. 180 brassica plants were grown. This is the first generation. The number of hairs present on the edge of each mature leaf was counted per plant and an average was calculated. The number of plants with the averages were counted and recorded. The plants with the most hairs, 25 or more on the leaves, were separated from the rest of the group and cross-pollinated with one another. The seeds produced by these crosses were germinated and allowed to grow into plants. 180 of these plants were selected to represent the second generation. The number of hairs present on the edge of a mature leaf from each of the second generation of plants were recorded. Here you see the results. Without artificial selection, average number of hairs, number of plants. With artificial selection, number of hairs, average number of hairs, and number of plants. Right. 321. Identify the independent variable. What are you controlling? If there is artificial selection or there is not. Okay. What are you measuring? As you can see here, you are measuring the number of plants with more hairs on leaves, but it's also here on the side here. The number of hairs was counted and there's what you're measuring. Okay, 322. State 2 ways in which the learners ensured that the investigation was valid. So, do you take this from the passage here? Because it said, ensured. So they used the same number of plants used in both generations. It was the same species of plants, same size of pot in which the seed was planted, same amount and type of soil, same amount of water was used to irrigate the plants, and same environmental conditions for the investigation. Give a conclusion for this investigation. So here you go to your results, and then from here you write a conclusion. So with artificial selection, there's more plants with more hairs on the leaves. So with artificial selection, can you see here? There's more hairs. There's more plants with more hairs on leaves, right? Okay, make sure you have everything included because that's quite difficult. You have to include everything from the table. Right, 3, 2, 4. State 2 ways in which the learners could have made their results more reliable. They could have repeated the investigation and taken an average, increased the sample size. 3, 2, 5. Draw a histogram showing the results with artificial selection. So only artificial selection. So you have the number of plants with the average numbers of hairs on the leaves, number of plants, average number of hair on the plant leaves, and there's your histogram, no spaces. The bars are the same width and there's no spaces. Then 3 to 6. Describe two differences between natural and artificial selection. So artificial selection, humans represent the selective force. Artificial selection is in response to satisfying human needs. Artificial selection may involve one or more species and it occurs over a shorter period of time. Natural selection, the environment is the selective force. Natural selection is in response to the suitability to the environment of the organism. It occurs within a species and it takes place over a longer period of time. You didn't have to draw a table, but a table is easier when it comes to marking. Right. 3.3. The diagram below shows the elongation of the neck of the giraffe according to Lamarck. 3.3.1. Use the example in the diagram to describe Lamarck's theory for changes in the giraffe's neck over time. So 3 marks. All giraffes had short necks. These giraffes frequently stretched their necks. They did this to reach the leaves that were available higher up on the trees. As a result, their necks became longer. The characteristic for long necks acquired in this way was then passed on to the next generation, and eventually all the giraffes had long necks. Any 3. 3.3.2. Why was Lamarck's theory rejected? Because there is no evidence to show that acquired characteristics are inherited. There's no evidence that structures that are used more frequently become more developed, or there's a change in phenotype does not lead to a change in genotype. 3.4. The diagram below shows one method of cloning sheep. So here you have the embryonic cell is removed from the donor sheep and allowed to multiply. 2. The nuclei taken from the donor cells and imported into foster eggs that has nucleolus ova from another sheep. They're allowed to develop. And 3. The eggs are implanted to the homes of foster sheep where they will develop until birth. 3.4.1. Explain why the lambs produced by this technique are identical to each other. Of course. Yeah. Because they contain the same chromosomes, because the embryonic cell has divided by mitosis, right? Or you could have said they are produced from the nuclei of the donor sheep, which divide by mitosis, or they have the same DNA. So any 2. 3.4.2. Explain why the lambs are not genetically identical to the sheep which produce the foster eggs, because the nucleus and genetic material originate from another sheep. Okay, the nuclei of the foster sheep were not used, so it did not contain the same genetic material, right? Or you could have said the ova used did not contain any nuclei, and it's the nucleus that contains the genetic material. 3.4.3. Describe how cloning in animals or plants can be beneficial to humans. Animals or plants with superior or favorable characteristics can be produced to enhance food production or biotechnology. And the last question, which is the essay, I still like to do the essay because it can be broken down into shorter questions and asked in your exam. Question 4. Describe Darwin's theory of natural selection and explain how it could lead to speciation by geographical isolation. So the first part, Darwin's theory of evolution by natural selection. Organisms produce a large number of offspring. There's a great deal of genetic variation amongst the offspring. Some have favorable characteristics and some do not. When there's a change in the environmental condition, or if there's competition, the organisms with characteristics that make them more suited survive, while the organisms with unfavorable characteristics make them less suited die. The organisms that survive and reproduce pass on the allele for the favorable characteristic to the offspring, and then the next generation will have a higher proportion of individuals with the favorable characteristic. Then, speciation through geographic isolation. A population of a single species becomes separated by a geographical barrier, and then you can give the example of the barrier, causing the population to split in two. There is now no gene flow between the two populations, since each population may be exposed to different environmental conditions or the selection pressure is different. Natural selection occurs independently in each of the two populations, such that the individuals of the two populations become so different from each other, they can become genotypically and phenotypically different. If the two populations are to mix again, they will not be able to interbreed. The two populations are now different species. This you get from your exam guidelines, grade 12s. Make sure you memorize it and you know it well before you go into any exams. Right. Thank you, grade 12s. Thank you for watching this video. I hope you learned a lot. Join me and watch all my other videos as well.